So I'll just give a quick outline of what we'll talk about today. That was a wonderful introduction. So I'm just going to very quickly introduce myself because sometimes I don't get an intro. And then I'll tell you guys a bit about nature methods. I'll tell you about our editorial process and our criteria for when we're considering papers. And then I'll switch gears to talk about more specific tips for publishing high quality microscopy papers. And then to end things, I'll talk about reporting and reproducibility in microscopy, which is the theme of this lecture series and also something that's just really important to me as an editor and as a scientist. So very quickly, my background is in microscopy, but I came to it from the probe side. I've been at Nature Methods almost 8 years. And before that, I was a postdoc doing live imaging of RNA using newly developed tags. And before that, I worked on the red fluorescent protein PS-Red, and you can see some of my engineered variants in this photo here. And I also studied chromophore chemistry. And the key takeaway from this is that I really believe methods drive biology, and that's also the ethos of our journal. When I was at the bench, I had 3 first author papers in Nature Methods. They're shown here. This does not make me a perfect judge of what should be in the journal, but it did really give me a sense of what the experiences is like working with editors at Nature Methods and what it's like from the author side to go through these papers from front to end. At the journal, I handle microscopy, spectroscopy, probes, and all the computational tools to go with it. A brief exception is a lot of the neuroimaging is covered by our neuroscientist, and we work together closely on those papers. I also get to handle chemical biology, cryo-EM and cryo-ET, and most imaging-based spatial omics technologies. I think this is a fun factoid. I have read over 4,000 submitted manuscripts as an editor, so I've been around the block a little bit, and I've gotten to read so much cool stuff. And I also wanted to plug this podcast series from Peter O'Toole called The Microscopists. If you want to learn a little bit more about me, I did a podcast with him, and honestly, mine is one of the least interesting ones. I think some of the other ones are much better, but that's a fun series to watch and listen to. Enough about me. I want to tell you about Nature Methods. And so we were launched in 2004, and we are now a team of 7 PhD holding professional editors. So none of us are academic PIs. We are all full-time professional editors. Nature Methods is a high visibility forum for publishing important new methods. Our editorial scope is focused on methods for basic research. And we are aimed at a very broad interdisciplinary audience of biologists. So even though we serve the methods development community, a lot of our authors are developers, our target audience is actually biologists. We place a strong emphasis on immediate practical relevance of the methods we publish. We like it for people to just immediately be able to pick up a method and use it. But of course, there's space in our pages for papers that have more of a conceptual advance and might have a longer tail in terms of influencing future work. And one thing that kind of sets us apart from some of our sister journals is that we actually don't need a new biological discovery to publish a paper. We have this sort of different bar, which is that we ask authors to show us that you can see something in a new way, or you can learn something new with your method, but you don't have to have really a big discovery portion of your paper. And this is our team. This is our chief editor, Allison Doerr, and Nina is our neuroscientist who I mentioned. Here's myself. Arunima is our proteins person. She has a computational background. Madura is our immunologist. Lei does experimental genomics. Lynn does computational genomics. The 5 of us are in New York, and Lynn and Lei are in Shanghai. And though they have the same last name, they are not related or married. And when I put it out like this, it seems like we're very distinct and we have, our own little areas that we handle. And that's true to some extent, but we all really work closely together. For example, I consider Nina my partner at the journal. We do all imaging and neuroimaging together. Lei and I do spatial omics together, among other things, like CRISPR-based imaging we work on together. Lynn and I do computational tools. Arunima and I do cryo-EM and cryo-ET. Medora and I do anything chemical biology related with antibodies. So really, and it's true for everyone on the team, really, it's a nice mesh work. And altogether, we have something like 35 or 40 years combined experience as editors. So we really work as a team to try to make the best decisions for the journal. So I just want to introduce you guys quickly to what it is we do. And I think that sets the stage for how I'm thinking about things later when it comes to reporting and reproducibility in microscopy. So what do we do all day? A huge part of our job is reading and evaluating new manuscripts. If we want to send a paper out for review, we choose and assign the peer reviewers. When the reviews come back in, we evaluate those reviews. And throughout this whole process, we are corresponding with authors, we're corresponding with reviewers, we're keeping everything moving. And of course, we see papers through the revision process and ultimately to acceptance. And that's for the research portion of our journal, which we also call the back half. Nature Methods also has a large magazine portion of the journal, which we call the front half. And that's where things like research highlights are, the editorial and other contents that we write are contained. We also do quite a bit of commissioning for things like reviews, news and views, perspectives, comments. Obviously, some of those pieces just get submitted to us, but quite a few, I'd say about half, are things that we seek out from the community and commission. In addition to that, we attend conferences and do lab visits and give lectures like this. So what is it that we do for papers? So the big thing I think is I like to think of myself as trying to bring a paper to the best version of itself. And that is, of course, within reason. We're not trying to make it a different paper. We're trying to make it the best version of itself. And that means bringing papers up to our standards. This can mean things like showing proper characterization or demonstrations. It could be just going through the workflow of doing our reproducibility checklists and making sure that everything is in order. It could be things like requiring data deposition. We guide papers through revisions and strengthen the claims. I'm not going to talk much about this at all for reasons of time, but we really, at Nature Methods, it's really our goal to focus our authors on the revisions that are going to move the paper forward versus laterally, and on the claims that are reasonable versus sort of like really kind of out of left field or really just way too much work or will make it a different paper. So that's something we pay a lot of attention to, and we try to give detailed guidance to our authors. Once a paper is accepted, we do careful line editing for things like clarity, content, and language. Our papers are also carefully line edited for grammar and style, but that is not done by us. That's done by professional copy editors who know all these little rules. Our production team makes the papers beautiful, and then once published, we offer high visibility. And I think this benefits both authors and readers. I think the vast majority of our authors are happy with the paper in the form that it's published, and they're happier with that version than the version that was originally submitted in terms of how robust the science is. And for readers, we want readers to be able to come to Nature Methods and trust that a method has been vetted, that it's been carefully looked at, that ideally they'll just be able to pick it up and run with it. And so I think the work we do helps both of those things be true. So that was sort of a nuts and bolts explanation of what our job entails, but I also wanted to kind of share with you more of a big picture. So what do we do all day? This is a job that doesn't just require us thinking about science and research, but we're thinking about other things. So what are important trends in science? How can we use the journal to highlight those? What do biologists need? What do biologists wish they could do in terms of experiments that there just aren't the right methods for? Can we help fill that gap? Are there areas or technologies that are just emerging, and can we shine a light on them? Can we help them get some momentum? Can we help them get some interest by championing them in our journal? We are always thinking about how we can improve the journal, make it better serve our readers. Are there things we can do to improve scientific publication? Can we make it faster, more transparent, more robust? Are there things we can do to improve science? You know, we have this platform. Can we make science more diverse? Can we make it more equitable? So these are sorts of things that make the job of editing really fun, and the sorts of things that we spend quite a lot of our time thinking about. The 7 of us are fully responsible for the scientific vision of the journal, and ultimately we're trying to create this curated journal that really serves you guys, that serves our readership. We want you every month to say, the table of contents is out. What did they publish this month? And I know that's absolutely a feeling I had when I was at the bench. I would read Nature Methods. I was the most excited to see the Nature Methods table of contents every month. And I would, sort of read it secretly hoping that I didn't get scooped by somebody, in the fluorescent proteins field or whatever. And I hope to give people that feeling that they're excited to see what we were excited about and what's been happening in the methods space. So just very quickly, I'll go through our editorial process, because if there's more junior researchers, maybe they don't know. So it starts with an completely optional pre-submission inquiry. And this basically tells you if a paper that you're working on fits us in terms of our editorial scope. And it's non-binding. So just because we say, yes, go ahead and submit, it doesn't mean we'll send the paper for review. But again, just because we say yes, it doesn't mean you have to submit your paper to us. Once you have your paper ready and you submit, we try to get our initial decision about reject or review within 1 week. And then if it gets sent for review, we try to have the first decision within 6 weeks. From there, for our papers, there's almost always a revision. And when it's resubmitted after revision, we try to get the decision to you within 4 weeks. Ideally, from there, it will go to acceptance, and that's actually the last stage of that the editor handles it. From there, it goes to production, where it goes to copy editing and layout, online publication, and print publication. And just because for lack of time, if you want more information on the career of editing, we did publish this nice editorial on it, I think 2 years ago now. So that's more our editorial process. I'm going to switch gears now and talk about our editorial criteria. So what are we thinking about when we read a paper? So the first thing is obviously the scope. Does it fit within our editorial scope? Does it match the type of papers that it's our intention to publish? And from there, it's like, how broad is the audience? Who is going to be reading this paper? From there, we think about ideas are, if it represents a big advance, and how significant, how potentially important the work is. We ask if there's practical value and if the method is generally applicable. Then we go into more technical details, like how well is it validated, how strong is the application. And although I consider myself the first reviewer of every paper I handle, I can't read absolutely everything for a very high level of technical correctness. We definitely rely on reviewers for that, but we are certainly looking at things like data quality. Are the controls there? Is it a challenging demonstration? These sorts of things are definitely things that we screen for at the editorial stage. And so for us in Nature Methods in particular, a big overarching question that we keep in front of mind when we read every paper is, does it enable new biological discovery? Can we learn something that we couldn't learn before? And the answer to this question has to be yes. So that's really our bar. You have to be able to learn something new. And so that's what we are looking for. And there's a few things we aren't looking for. So one is reinventing the wheel. And by this, I mean a method that takes you to the same endpoint as an existing method using an alternative strategy. So we tend not to publish those papers. We tend to not publish small tweaks to a protocol, but this one has some notable exceptions. We published this paper where an improved buffer allowed you to do DNA paint 10 times faster. And because speed is such a bottleneck for DNA paint, we thought that even though, conceptually it's kind of a small tweak, practically it was probably important enough to review. So this one has some exceptions. We don't publish nice applications of established methods. So by this, I mean, let's say you have a light sheet microscope and you're going to be the first person to image some certain structure or small organism. But if the method is established, we tend not to publish just the application of it to some new sample. We don't publish methods without a strong biological application. Again, our readership is biologists. We tend not to publish cheaper alternatives to established methods, although again, there are some exceptions here. And we don't publish methods that are primarily for drug discovery, diagnostics, or clinical applications. And that, again, that's just because our scope is focused only on methods for basic research. And before I leave this slide, I just want to say these are all important categories. So we are not making a judgment about whether it's important to do this kind of work. It's just not the focus of our journal. In fact, other journals publish these papers better than we do. And so that helps us limit what we're publishing. And I always like to mention Nature Biomedical Engineering. It's a newer journal, although at this point it's pretty well established, but they publish a lot of methodologies that are sort of in the same method space, but the target is much more medical or clinical or diagnostic than what we will publish. And I also like to mention here that because of our focus, we published almost nothing on COVID-19 during the past 3 years. Not nothing, but I'd say fewer than 10 papers, whereas other journals were completely swamped, have issue after issue of COVID-19 papers. It didn't match with our scope. So I think that's something that's also sort of separated us from some of our sister journals. So a lot of people ask me about the odds, so I just included this as a slide. We get around 200, well, over 200 new submissions per month. It's rare that we'll have a month with fewer. We review 10 to 15% of those, and of those, 60% are ultimately accepted. I think for me personally, I am on the higher end of this, and I think I'm closer to 70% of the papers I send out getting ultimately accepted. But this does vary a little bit from editor to editor, usually because we have different levels of interest sort of in different topics as time goes on. But that's generally our statistics. So it's pretty exclusive, but not impossible. So that's enough about our editorial criteria. Now I want to switch gears into telling you more sort of specific tips for improving methods papers. And for this, I think a lot of it is generally applicable for methods papers, but I did put an emphasis on microscopy here for you guys. So the first thing that I cannot emphasize enough is that for Nature Methods paper, the method is the star. The method is the result. So it shouldn't be buried in the methods section. It shouldn't be skipped over to go straight to the biological findings. The method is the result. So it really deserves to be center stage. And just starting things off, we'll sort of go in order through a paper a little bit here. You want to start just by placing your work in context. Doing this well goes so far to making editors and reviewers happy. Tell us, what's the state of the art? How does your method move us beyond the state of the art? And why is this advance important to the audience of your target journal? So in our case, biologists. And here, again, I can't emphasize this enough, really explicitly state the conceptual advance, like what is happening here that's new, or practical advance if it's more of a practical paper. So then benchmarking. It's really important for a methods journal to show direct comparisons, if at all possible. And the idea of this is not to like poo-poo on other methods or other papers. Everyone is trying to improve. The idea is not to trash what came before you, but rather just to demonstrate the extent that your method improves data acquisition or analysis. The idea is here that you want to convince a reader to overcome the substantial activation energy associated with trying something new, especially if what they've already been doing in their lab works pretty well. So, you want to think about convincing, what would it take to convince a developer that it's worth building off your approach? What would it take to convince a biologist that it's worth switching over to your new method? And those are sorts of things we look for as an editor, like who's going to want to do this? Who's going to, for example, if I get a new fluorescent protein paper, I think, is it going to be worth people to reclone everything they have labeled with M. scarlet to now look at everything with this new red fluorescent protein? Is it sufficiently good that people are going to take those steps? So that's the sort of thing I keep in mind when I'm reading a paper. And the more you can show it, the better off you are. And again, the method is the star, so it's really important to provide quantitative characterization of your approach with pros and cons. what's the spatiotemporal resolution of your method, and really use good metrics, use FRC, use rulers, use, standards, these sorts of things, quantitative comparisons, really quantitative metrics really make your paper look strong. For a microscope, how deep can you image? These things like this, what's your depth of field, what's your practical limit? These sorts of things are things potential users want to know, and it's really important to characterize. How sensitive is your instrument? And sometimes, what are the optimal samples? Is your instrument really only suited for cleared tissues? Can it be used on things that have more scattering? So really characterizing these things are important for our papers. It's really important to show what you can do. And I'm going to have a whole other slide on this, so I'm not going to spend too much time here. But we expect challenging demonstrations that showcase the potential of the method. And if it's something that the authors are really pushing as being generally applicable, we ask for that to be shown as well. This is super important, especially for methods papers. Really do the work to make it reproducible. And that means different things for different papers. It could mean sharing a detailed supplementary protocol, which we often require. It can mean sharing code. It can mean sharing source code. It could mean sharing your test data. It could mean just very, very detailed notes about how to build an instrument or schematics. But this is something that's really important, especially for methods papers. And I'd say, as you look forward when you're writing the discussion, you want to explain the big picture. Say, okay, down the road, I can envision such and such and such applications of this technology. But here, just be a little bit careful that you don't say, oh, it should be immediately applicable to this, 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 because then you might set the expectation, in the referee's eyes, like, well, if you think it can be, why don't you show it? Because you want to just, there's a fine line there in overclaiming more than you show, or explaining the potential of a technology. And this, I think, is super important, especially if there are any early career researchers watching today. Is just read your target journals. Do they publish on your topic? Do a keyword search. What have they published in the last 5 years, in the last 10 years in your area? If it's nothing, there's a possibility that it's not a good fit for the journal, probably because of editorial scope. If you do your keyword search and you pull up a bunch of papers, how are those papers structured? What did they have to show in their papers? Are there any standard experiments or things that most of these papers have that your paper doesn't have? Try to look for these sorts of things. And then, like I said, I was going to come back to demonstrations. This is just so crucial. I would say this distinguishes the good from the great papers that we see regularly, every day, I'd say. So, as I said, our readership is biologists, but we serve the methods developers community. And there can be this disconnect between developers and biologists. And this is a good thing. This is a great thing. It's not the job of every developer to imagine every downstream application of their tools. But that being said, you can't assume it's obvious why your approach is needed. Let's say you have an instrument that images 10 times faster than the state of the art. But if the state of the art can already image the fastest process in a living cell, then what's its importance to our readership? Why are we going to be interested? You have to fill in that gap. So then the best demonstrations really showcase the benefits of your method, again, for enabling new discoveries. With a microscope, what can you see now that you couldn't see before? What can you track now that you couldn't track before? In the best case scenario, the demonstration will speak to a broad audience. Maybe everyone in cell biology, everyone in neurobiology might have an interest in your findings, or developmental biology. Try to think along those lines, or are relevant and especially timely. And here, this is super important. You really want to back up the claims you made in your paper. So, I see papers where 90% of the paper is talking about live cell super resolution imaging, but then the final figure is on fixed cells. And so they didn't, they're saying this is an important advance, but they didn't show that it got you to to the end. So think about that when you're planning your demonstration. And sometimes it can be a matter of, just how you pitch your paper. But if you're going to make your method about live cell super resolution imaging, show us what imaging a fast process looks like. This is just one example. And is your method generalizable? If you really believe that it's broadly applicable, then show us on samples that are not too similar. Don't just show us on 2 different mammalian cell lines. Maybe show us in vivo versus in eukaryotic cells, or on a fish, or think along those lines. And again, if in doubt, go back and look at what we've published. We have a big training data set available for you to really get a sense of what our standards are. And also, you can talk to us as well. If you are an early career researcher, especially, you're not sure if your story is something that we'd be interested in, shoot me an email. I'm very happy to chat. So then I'm just going to give a few examples of things that kind of show what I'm saying, because I know I'm a pretty visual learner. So this paper is the Mesospin paper. You don't have to understand the technical details, but this is an axially swept light sheet microscope, and what it offers is very good isotropic imaging resolution. So what you're looking at is a cleared mouse brain with Thi1 YFP labeled. And what they're showing you here is very rare in microscopy, an XZ view. So what you're seeing here is if you image with a standard Gaussian beam, you'll get this blurring along the optical axis that's very typical, often hidden in microscopy images. Whereas if you use the Mesospin, you'll see these nice isotropic resolution, these beautiful cell bodies. So I think immediately they have a challenging sample. It's an intact mouse brain. It's something of broad interest. Lots of people are interested in functional and structural connections in the mouse brain. And they're showing you side by side what you can gain. So if I'm working on segmenting cells in the mouse brain, it's very clear to me which data set I'm going to want to start with here. And then again, beautiful XZ view. They show us a zoom in where they can see the fine neurites, both in XZ and in XY. And so I just love showing this one. If you've seen me talk before, you've probably seen this before, because I just love this figure. But I will say this isn't even the most beautiful figure in this paper. So I'm not just cherry picking. Another paper, again, I think is nice. This is the Decode paper. This is a fast single molecule localization microscopy paper. And what they do here is they use deep learning to fit frames with dense emitters, so you can do faster live cell super resolution imaging. I really had to push the authors here to do a live cell demonstration, but they did. They ended up showing me live cell imaging on 2 different structures, changing longitudinally and on NUPs. So I thought this would end up being a really beautiful demonstration. And then they show, some of their more standard fixed cell imaging as well, and what they're able to achieve in the Z dimension. So I thought that was a paper that has really nice demonstrations showing what you can do. And then here, this is for those of you who work more on the computational side, image analysis side. This is the No New Units paper from Klaus Meyer-Hein. I think it came out probably a year ago, but essentially they were speaking to this debate of whether you want more and more complicated or new ANNs to do image analysis, specifically segmentation, or if you could just optimize a UNet and get comparable results or better results. And what they showed here is that they have this way of optimizing UNets for the task, the segmentation task they're interested in. And if they do that, it essentially performs at best or near best of many, many different large-scale imaging competitions. And when they submitted this paper, they had already had something like 20 demonstrations, but they were a lot of medical imaging. And I said, okay, well, we like this paper, but you need to show us more cell biology or neurobiology. So they added some demonstrations for us. And again, for a reader of Nature Methods, they're going to go to this paper, and ideally they're going to see something that looks quite similar to their task and say, aha, this is going to work for what I need to do, because they showed me on all these different examples. So I love this paper as well. Okay, switching gears. I'm good on timing, I think. So then I told you about what we're looking for, what we're excited about, and how to show it. And now I want to switch gears and talk to you guys more about the theme of this lecture series, which is just reporting and reproducibility in microscopy. And this is something that's near and dear to my heart. We actually had a focus issue on this exact topic last December, which took well over a year to put together and to coordinate. But I think it just came out so beautifully. And I urge you all, if you really have an interest in this area, to read these pieces. And I'm going to go through a lot of what the material that's in these pieces in the next few minutes. But I'm so proud of this. I think it's so interesting. So first off, how good is microscopy reporting? Is there a problem? Or are people doing it right already? So there was this really fantastic paper, not the first of its kind, but it got a good amount of attention. It came out in eLife 2 years ago. And basically, these authors looked at 240 papers in 8 different journals and just said, how good is the microscopy reporting? And long story short, not very good. So here is a table summary of some of their results. These are the journals. This is the percentage of the figures that included microscopy images. They didn't count Western blots or other gel blots as images. Then they looked at what fraction of the methods section was devoted to describing the imaging. So look, 80% of their data was images and 4% of their methods was describing the imaging. So, not ideal. And then they had this sort of binary test of, did it pass a methods quality test? And there were 3 very easy points on this test. Information about the objective, so like the NA and the objective type, I think, then the voxel size for the images, and then excitation emission information. So did it include those 3 very simple, really reporting facts? And essentially a lot of journals did really badly. I feel I haven't done this independently for Nature Methods, but I read for those things, and I'm almost certain we would be closer to like 100%. But I can't say that for sure because I haven't done it. But it doesn't surprise me that, for example, Biophysical Journal did very well, relatively well, because a lot of their authors are developers, are microscopists. But this paints a really bad picture, right, of what the state of the art is in microscopy reporting. And so there's definitely a need for, and it's not, I don't think it's anything the authors are doing to try to deliberately mislead. There just might be a lack of standards, a lack of guidelines, and a lack of understanding of what ought to be reported for reproducibility. And so if we bring those things to light, we could really improve the quality with relatively little work, I think. So let's see if we all agree. So I don't need to convince this crowd probably, but why are reporting and reproducibility important? Just in the world and also to us as editors. So this goes without saying, but reproducibility is necessary for scientific progress. Robust and repeatable scientific conclusions serve as the strongest foundations for future work. So clearly you want to build your work off of something that is reproducible, right? And just, from a very much more narrow viewpoint of an editor, we want authors to be able to reproduce the work that's published in our pages and to replicate the studies independently. And even beyond that, even beyond replication studies, we want these methods to be applicable to distinct biological questions. And so that is a really high standard for reproducibility. And this is just so crucial, especially for methods papers, because methods, we've seen time and time again, methods that can't be implemented elsewhere, they basically have limited value apart from, they might have an initial splash and then sort of just become invisible. It's really these reproducible methods that take off and become impactful and become useful across biology. So this is very important to us to screen for these to the ability that we can using peer review. And so how is reporting connected to reproducibility? Reporting is crucial for ensuring that generated data could be obtained in another lab, that the analysis of the shared data produced in that study could be repeated to reach the same conclusions, and that an approach or device or instrument described could be replicated. So the way we make things reproducible is sharing how we did them in a really detailed way. And it's really important to state here that proper metadata tracking and reporting improve data reuse as well. So, when you're using microscopy for a specific biological question, your data might really just speak to that point. It might only be important for one study or a set of studies that are closely related. But we are in the age of big data, and big data has come to microscopy in a big way. Even just in the 8 years that I've been an editor, we've seen a huge rise in atlas projects, the brain, the kidney, every cancer. So I think as data reuse becomes more and more important, metadata tracking and reporting becomes just essential. It's just really completely necessary for people to reuse data outside of the lab it was generated in. So I hope I've convinced you that it's important. So then the question is, what is necessary? So we need better metadata tracking and reporting. Metadata is the data behind the data. So if you see an image, a beautiful image in a paper, what did it take to get there? Sample and sample prep details. What microscope? What were those microscope specifications? What were the acquisition parameters? Did you use any analysis? Did you use any image augmentation software? Did you use any analysis software? All of these details are really crucial for reporting. And in the issue I just highlighted, we had 3 comments, these 3 pieces from, so this one comes from Paula Montero Lopez and colleagues, and also includes Thomas Pango and Mark Sanders, who were authors on that eLife paper I discussed with you guys. And it really puts forth best practices for reporting reproducible fluorescence microscopy methods. And Paula runs a big core facility at Harvard. Michelle also runs a big core facility, but I forget where. Lisa as well. So the authors on this have a lot of experience with different modalities and with what it takes to write a good method section, or to even try to repeat an experiment that someone else did. This piece comes from the Bioimaging North American group and describes metadata standards for light microscopy. They follow the OHM model. REMBI is another group. I think this is a global group. It includes global bioimaging. They have recommended metadata for both light and electron microscopy. And then in a separate issue, the Human Tumor Atlas Network and HubMap people came together and gave us guidelines for highly multiplex tissue imaging. So as you can see, this is a really timely issue. A lot of people in big consortia are saying, okay, now is the time to really come together and make some good guidelines for people to do accurate, high quality reporting. So what have we learned from this at Nature Methods? What do we require? So some of this is not new. It's not necessarily changed due to these guidelines, but we've always required, strong methods sections. So in terms of sample, what cell line, what strain you used, how you did your sample prep, how you did your image acquisition in terms of your microscope and settings, any image processing done, what software you used, what modifications were applied, and any image analysis that you use to get your quantitative results. So this sort of thing has always been our standard and will continue to be our standard. And it goes beyond that. So we certainly have papers that use microscopy data to support some sort of conclusion, but that's not the typical for us. It's more typical that we're having a new modality. And so for these, we need a full description of the system design, ideally with schematics, freely available code that we get tested by the reviewers, and freely available raw data or test data for the referees to test with the code. So these are things that we're also requiring. And so will it get more strict than that? I think we're probably working towards some sort of checklist or table for anyone using microscopy to fill in as a requirement for publication. but we're not quite there yet. And I think we're just sort of waiting for consensus to grow among microscopy communities. You know, the guidelines we publish, they're not all identical to one another. Some are a little bit more strict and some are a little bit more flexible. And we can talk more about why that might be. But something I'd say that's really important in Nature Methods is that we want to strike this balance between maximizing reporting without creating an undue burden on researchers. We don't want this to be some sort of miserable drudgery, right? We want this to be something that becomes second nature. Every time that you do a microscopy experiment, you remember to record this, and this, because eventually when you publish, you're going to wish you had it. So that's the sort of thing we want it to become sort of default with lab note keeping. And again, in that focus issue, I told you I'm going to go through the highlights. We publish tools to make it easier for both for biologists, but also I'd say people working at core facilities. And so we published Endemic, which offers flexible editing of microscopy metadata, Micrometa app, which is for user-friendly tool for extracting and collecting microscopy metadata, and Methods J2, which is sort of the one I love the most conceptually because it helps to capture your metadata and then automatically generate a template methods text. So it's sort of, here's your metadata, I've spit it out into a methods text that you can then fine tune. So I think that's just lovely. And these three tools are designed to work together. In the perspective from Montero Lopez, they also developed MicCheck, which is an interactive spreadsheet for metadata recording. And I would say that, you know, as instruments get more sophisticated, it's becoming more common for biologists to work with core facilities. And I think that's great. And these experts, you know, can really help with these sorts of metadata tracking and methods reporting. And I think it's good to lean on these experts, you know, with a lot of microscopy expertise. And these are also the people who are helping develop the standards. So it's sort of full circle. Here, sorry for the bad resolution on the screen grab, but I wanted to say that that comment the perspective I was just mentioning from the Montero Lopez piece, the extended data figure 1 is a poster that's a visual guide to metadata reporting. And I think a lot of people like to print this and have it by their microscope. So I just wanted to tell you guys that that exists. I'd say what's on our wish list moving forward. I'm almost done, so I'm wrapping up. You know, we would like to have this fully complete metadata reporting checklist that just ensures the quality of all the microscopy reporting in our papers. When we publish new microscopes, we would love to see CAD files of the designs come with them. We would love to see fully open source code. And I only call out MATLAB here because MATLAB is the most frequently closed source code that we see. Yeah, okay. So I think we were all on this slide. So how do we ensure the best reporting? If you've published with the Nature Family Journal, you've seen that we have various reporting and statistics checklists we ask authors to fill out. These include a lot of what would typically be considered metadata, like cell lines used, antibodies used with, you know, model with catalog numbers, and to list the software used for data analysis. We also have specific checklists for certain types of experiments, like if you're using microarray data, there's a specific checklist for that. If you're doing structural biology, we have a crystallography table, a cryo-EM table, and NMR table. We don't have one for microscopy yet, but I think moving forward, that's something we will have. We ask all authors of all papers with substantial microscopy data to upload to a repository. We ask for all reviewed code to be uploaded as supplementary software so that it's maintained with the paper. We ask referees to test code and software that's provided. And to make this easier, we have a long-term ongoing trial with CodeOcean to make Dockerized or containerized versions of code for review and also for posterity. And, you know, a thing we do is we just ask the referees, could you reproduce the experiment or the data based on the details reported? And very often they ask for quite a lot of clarification. And, you know, it might not be obvious to the authors what's confusing, but the review process really strengthens that. And, you know, sometimes people say they'll make their data available upon request and they don't. And so if that happens to our readers, they can reach out to us and we will work with them to reach out to those authors and try to get that to the best of our ability. This is my last slide. So what about the challenges? What about the future of this? So I think a big challenge is that microscopy data are huge and getting huger. You know, people are dealing with multiple terabytes of data from like a single experiment or from a few experiments. I think we need to have better ways of dealing with big data. That's not a problem with metadata reporting necessarily, but it is a problem for reproducibility and reuse. I think there's an ongoing question in microscopy that is not a great topic for today, but it's interesting to chew on, is what exactly constitutes raw data? What do we need to be storing? What do we need to be sharing? I think that's an interesting topic that will affect this community. You know, storing, hosting, and sharing these data are becoming more and more expensive. Who's going to cover that cost? What are the minimum needs for sharing and reuse? I think a challenge for metadata tracking and reporting is that it varies so much across modalities, even just within fluorescence light microscopy, you know, it varies. So I think that's an ongoing challenge that could be addressed. I think, you know, I love the idea of image repositories as places where you can just sit your raw data and people can access it as they please. But although the existing image repositories and resources are fantastic, I'd say they're not yet meeting the needs of all microscopists. So what can we do to grow that area, and what needs to happen moving forward? There are a few things in play at Nature Methods working with Figshare, but I can't talk about them fully yet, but they'll probably be discussed in future editorials. You know, I'd say that microscopy has always been sort of pioneering and open, but we're definitely part of a cultural shift towards open science and data sharing, and some aspects of that lag behind. So how can we catch up? And I mean that, like, you know, even me when I was at the bench, I would not have expected to have to share, to have saved and stored and filed away all of my imaging data that I ever took in such a way that someone besides me would dig through it, because why would they, right? Why would they want to? But that's sort of, this idea is sort of changing. How can we make things more available and more shareable? I think, again, it'd be great to reach a consensus on metadata requirements. It'd be great to have those out in the field and see if it actually improves reporting. Like 10 years from now, or 5 years from now, can we do another study like that one in eLife and say, look, we implemented these changes, these guidelines, and look how much better reporting has gotten. And then will that subsequently improve reproducibility and reuse? I hope so. And then I just wanted to mention that, you know, I'm excited about next generation microscopy file formats and what they can do for improved versatility and improving things like metadata tracking and data sharing. That's it. Again, I'm so sorry for the technical difficulty, just bad luck. But thank you so much for inviting me. Thanks for listening. I would love to get to know you guys as part of the Latin American bioimaging community. It's not an area I know well, and I would love to know what your pain points are, what you're excited about in terms of methods development, and just get to hear about your work. So if anyone wants to reach out and have a one-on-one chat with me based on this talk, I would love to hear from you, and I'll take any questions.
Thank you very much, Rita. Great seminar. I enjoy a lot, and I think you cover basically all the different, you know, edges of publication, from the formalities of editor and the editorial processes, what is required to, you know, publish a method in Nature Method, right? A new paper in Nature Method, and also, you know, this last part of, you know, how the journal is helping to share, you know, the work for the community in terms of reproducibility and you know, good practices for methods is very important. Some of the papers that you have mentioned were some of the speakers that we have in the seminar list. So Michelle Aitano, Caterina Strambio de Castilla, Claire Brown. So they were all, you know, part of this fantastic work. And yeah, I think it's a really, really important point for, you know, our community. And in my opinion, our show, you know, we are a core facility that blends between, services as well as develop some tools and techniques in terms of microscopy and imaging. Part of our shop is also to help to our users to improve, the papers and have, or stick to the good practices that we are trying to develop today. So we have the chance for asking questions to Rita. So for everyone who would like to make a question, you can paste in the chat, as I said before, or you can raise your hand and we can open the mic. I have some question and some comment to make, so I can eventually roll the eyes. So you hit at the end of your talk, all these different aspects of improve our best practices in reporting data method. And one thing that came up during this discussion, and it's very nice to have your opinion, is exactly what you say about repositories and sharing the data, raw data, sharing the code. Because one thing is, what is true is, well, I think I absolutely agree, it's unacceptable, or it's not unacceptable, but it's not really fair to say, well, I will share my data upon request, right? It will be nice that this will be available. But there are bottlenecks, right? Where we are going to deposit. In my last 2 or 3 papers, I try to start to share basically data, but we always struggle. I don't know, figure share or some other repositories that you don't know where you are depositing. So how the community, journals, some global initiatives, etc., can work together in establish, right, a proper repository where you trust where you put in your data, when you trust where you are depositing your code, whatever. What is your thought? What is the thought of Nature as a big journal in that sense?
That's a great question. So, this is one of the reasons we can't demand people share all of their raw data, because there's no, equivalent to GenBank or to the PDB, right, for microscopists to use. That's just not, there's no infrastructure for that right now. And like, there's the image data resource, there's all the stuff that's happening at EMBL for data hosting, but it's just not enough volume. And microscopy data are huge, or they can be, especially raw data. I don't know, I would love to see, gosh, I think we could tackle it a number of ways with the tools we have now. There's Zenodo and Figshare, but I think they're not free. So I would love to see journals.
Depending on the size of your data, it can be or not free. But
yeah,
if
exactly.
you have oversized it, you have to pay. Absolutely, yes.
Right. So I feel like I honestly would love to see that be something that journals take on. Like, journals should take on the cost of hosting the raw data they're publishing. I mean, I think that's at least everything directly related to the manuscript they're publishing. I think that's one option that's actually not unfeasible, because it's companies working with other companies rather than having the authors take on that burden. But what really needs to happen is that governments need to say, look, we need to have the infrastructure and the commitment and the investment to host and maintain long-term imaging data, and not just from Atlas projects, not just from these big, well-funded projects, but also, from individual labs. And I think, how to get there, that's a good question. I actually haven't spent that much time thinking about, but maybe, maybe an editorial, maybe some campaigning could be done, because I think the time is right for it. And you don't want to have to piece together a solution to what is ultimately a fairly universal problem. Absolutely. But I think that's what's going to take.
It also has different edges, because what you say also at the end is, well, okay, we can deposit files, but how we have, a consensus of, which kind of file, which kind of, metadata, how is the, there are every microscopy brand changed from the from the size of the screw of the objective to the way that they save the metadata. So it's very complicated, for individuals to think on that. I think it's really need to be elaborate between industry, between journals, between government, as you say, but also other organizations, right? Philanthropists and global organizations like GBI, working groups, on these kind of things to reach at some point some sort of consensus, right?
Yeah, absolutely. That's all going to be really important. Interoperability is, I didn't bring up the FAIR principles on this talk, but I think probably everyone is familiar with them. But just interoperability and engaging with the data, that's it's going to be challenging. I like that you mentioned companies, because you're absolutely right. Companies are going to have to be a big part of this buy-in as well when it comes to these sorts of details.
Even when there are parts from custom built things to full commercial. I mean, in the middle you have a lot of intermediate, like the companies for the detection, the companies for moving whatever, scanners, etc. There are a bunch of information there that need to be at some way or another good documented in order to be reproducible. Otherwise, it's really, really hard, right?
Yes, absolutely.
I have another question, but it's more general about nature method. So far, I recognize the brief communication, the full paper, and some other like editorials or comment, and I crossed over some of your Twitter activities, new ideas of like comparison between like new method with other standards or something like that. Can you briefly at least comment of the different goals of the different publication that you receive in the nature method?
Yeah, of course, I'd be happy to. So let's split it up into a few areas. So first of all, there's like front half, we have reviews, which are supposed to be really in-depth look at a relatively broad field, very scholarly, very rigorous. Perspectives are similar. Their perspectives are often very review-like, but they're typically on a smaller or newer area. So not as far-reaching as a full review. And then also in that sort of area, we have comments, which are, these are opinionated pieces. So they might have some scholarly portion, like this review material, but they usually come to a conclusion, or they have a strong opinion that they're trying to share. So those are front half formats. And if you don't know exactly the right way to submit, just submit what you think is best. And that's the sort of thing we figure out. We're like, actually, this is best as this format. This is true for all of this. So then in terms of research articles, which you guys are probably maybe more interested in. So there's the brief communication. So this has actually changed a bit in the past since we have our newest chief editor. We use brief communications. Really, it's supposed to be a really brief report of a new technology. It doesn't necessarily have a big or any large results component. It's really just sort of a straightforward advance. This is your figure showing it works, and it's supposed to be, it's like small and self-contained. The vast majority of the papers we're publishing these days are articles where they'll have, much more demonstration, much more benchmarking than a brief communication. But that's pretty much what separates the two. Usually a brief communication is just like a quick idea. It works really well. We're showing it works really well. Whereas an article is a more fully developed story, often has more benchmarking, has more demonstrations. And if we push a brief communication to demonstrate more, then we'll just change it to an article.
Okay.
So there's fluidity between those two formats. But then you were talking about the comparisons. So this is something that's really cool. We have always published analyses. An analysis is just a format where you're not describing a new tool. You are just comparing.
Okay.
So it's just fully benchmarking, but nothing new. You're saying, okay, there's 10 algorithms for doing single molecule localization. We're going to compare, actually there's way more than 10, but we're going to have a competition. We're going to compare them all, see what works the best on different types of data, and then we're going to write that up as a story. So that's an analysis. And we've always published those, and we still will. Our newest format is actually a registered report. In a registered report, what you do is you submit a plan for a competition. Say, I plan on comparing these 10 tools on these 3 data sets. I'm going to do such and such analysis, and I'm going to, and the goal is to come out with which of these software performs the best under what conditions. You submit the plan and maybe some preliminary data, but not necessarily. We get that reviewed and revised. And once that plan is signed off on by us and the reviewers, that paper is actually accepted in principle. Then you do the experiments. And so long as the experiments are done according to the plan, no matter what the results are. So let's say you're comparing 10 fluorescent proteins, and no matter which one actually ends up performing the best, as long as you did the experiments as you described, and it's done all in an unbiased way that the peer reviewers agreed upon, we will publish your paper. So that's a registered report. And we are trialing this for a bunch of different competition papers, because one problem we would have with analyses is that the reviewers would say, oh, it's a nice idea, but they used all the wrong test data. This is the wrong analysis metric. They should have done it like this, this, and this. And by the time the competitions run, you can't change anything. But if you do it as a registered report, you get the feedback early, you can get a much nicer paper.
Absolutely, yes. It's a very good point, because otherwise it's like, it's really frustrating when you already, embrace something and you go in the wrong direction. Absolutely, I agree with you.
So we have a few of those in the pipeline that I think should be really good.
Nice.
We'll see.
And if you have to tell, what is the main pitfall that usually you see when people is preparing, paper for nature method? What is your main, main error that people make when they prepare? I think that what you tell today about what you expect about a nature method paper, which is the method is a result, is a really strong statement, right? Because most of the people always circumvent around, one experiment to demonstrate this, one experiment to demonstrate another, but you don't hit the point. If you are, having a new microscope, this is your result. It's a new microscope, which is a different conception. But what is the main, mistake people does?
That's a great question. So I would say there are 2 big ones. One that will get a paper immediately rejected is just being outside of our scope, like too clinical, too diagnostic. In those cases, it's like, oh, did they even read the journal? Did they even read our scope? So I'd say about 10 to 20% are just quickly eliminated in that way for being out of scope. So that's the biggest mistake, but that's a very quick rejection. In terms of papers that we would consider, the biggest mistake is not going all the way to a challenging demonstration. And, so they often stop at proof of principle. So it's like, okay, my method should do this. I'm going to show you on a biological specimen that it does do it. And then it just sort of ends, versus going all the way to that next step, to pushing the method to its limits, showing that you can do something that's actually very hard. So I'd say that's the biggest difference between a good paper and a great paper, is that what we would call the 4th figure. And that, this is a challenging one, because this is a case where labs with a lot of resources, labs with a lot of connections, a lot of collaborators have a big advantage. If you're working someplace like Genelia, and you have people working on flies, working on fish, working on this, working on that, just right around the corner, and they can, plug your tool right into their system, and you can have this cool demo 2 days later. there's a real advantage to that. And I think that's something we're mindful of, but I would say that going that extra mile to showing not only does it do what it says, but you can look at something really exciting now. That's something that really separates our papers from what we see. I would have to say. But I'll also say this: if we love a paper, we'll often say, okay, we're going to give you, we want to review your paper, but you're going to have to add, but if we're eventually going to publish it, you're going to have to add such and such demonstration. I'd say this comes up for me a lot in cryo-ET, because a lot of people submit me a paper and they just show on the ribosome. Because that's it's gold standard. Everybody understands what a result means on the ribosome. And I'll say, look, I love your paper, it's fantastic, but you're going to have to show me something in addition to the ribosome. And I'll say, I'll review it as it is, but in the meantime, while it's out for review, you need to be working on something. And sometimes they'll tell me I can't, which tells me something about the general applicability. But usually they say, okay, Rita, we'll work on something. I'll do also the proteasome, or I'll do also something else. So that's one example. I can think of millions of others, where I just say, look, I'm going to push you to show me what you can do. We published this. Oh, you're from Enrico's lab, so you know phasers. We published this hyperspectral phasers paper from Scott Fraser's lab.
Yeah.
And the initial paper just showed 3 color imaging, and I was like, look, this is amazing technology. You're going to have to show me what you can do. And then they ended up coming back to me with 14 color imaging and an intact zebrafish, and I was like, this is what you could do. This is what we wanted to see. So that's the sort of thing we're really pushing our authors on. And that's something that's really important to us.
Yeah, absolutely. Yeah, I agree. Yeah. I think one of the things that sometimes happen is that you don't sell in the right direction all the potential of the tool, and this is a big fail usually, because you say, well, I cannot understand how others cannot see this potential, but at the end it's your fault because you don't do the proper advertisement or the proper examples to demonstrate all the potential. And yes, this is I agree with you. This is one of the big mistakes. So I don't know if there are any questions from the audience. We are a little bit over the hour, so if not, I would like to really thank Rita for this time. I enjoy a lot. We will upload the record to the YouTube channel of the Advanced Imaging Machine Unit here, Pasteur, and we keep in touch, and I hope we can submit sooner or later some document to Nature Method too.
Yes, absolutely. If anyone in the audience has a paper pitch for me, please just email me. I would love to hear from you.